I reckon we'll get started in a second. Just a quick note before we do get started. If you have any questions, DM any of us and we'll pop them on the questions doc, which will be a good revision resource for you. And this will be recorded and uploaded to the YouTube channel afterwards. So don't worry about trying to take everything down because you'll have the slides and you'll have the recordings later on. Meg, do you want to start us off?
So we've got quite a bit to get through today. It was quite a big week of physiology. So we'll just jump right into it, starting off with resting membrane potential. So these are your learning objectives. Describe the processes for electrical charge in the semi-permeable membrane. Apply the Nernst and Goldman-Hodgkin-Katz equation for calculating resting membrane potentials. And describe how a semi-permeable membrane can create a difference in electrical charge and how this can be applied to the nerve cell membrane. So first, what is resting membrane potential? So under resting conditions, which is basically all the time when an action potential isn't being generated, which we'll discuss next. So this is just normal conditions essentially. Cells have a potential electrical difference across their plasma membranes. The inside of the cell is negative and the outside of the cell is positive, as you can see in this diagram here. This is due to differences in the concentrations of ions inside and outside the cell, as the plasma membrane is selectively permeable to different ions. So if we go to this slide, the extracellular environment is composed of mainly sodium and chloride ions. As you can see, we've got the pink sodium ions and the purple chloride ions. And the intracellular environment is mainly composed of potassium ions and organic protein anions. You don't really need to know anything specific about what these protein anions are, just that they are negatively charged. Overall, neurons have a resting membrane potential of negative 70 millivolts. So that's an important number, which you need to remember. And we're talking about the inside relative to the outside here. So because the inside is more negative than the outside, that's why it's negative 70 millivolts. So there are a few factors which create this resting membrane potential. We've got concentration gradients. So that's the fact that ions will diffuse down their concentration gradients from an area of high concentration to low concentration. Selective permeability, that's the fact that the membrane will allow some ions to pass through while others are stopped. And finally, electrical attraction. So that's just like a basic concept from chemistry, how if you have a positive and a negative ion, they will be attracted. Whereas if you have two positives or two negatives, they will be repulsed from each other. Types of transport channels. So now we're talking about types of transport channels which are embedded in the plasma membrane. These are all like your protein transport channels, which might be familiar from biology. So there are 4 different types. We've got mechanically gated channels, voltage gated channels, ligand gated channels, and leaky channels. And you'll find these just studded throughout the plasma membrane. So first we've got our mechanically gated channels. So these open and close in response to physical changes in the environment, including touch, pressure, or temperature. Then we've got voltage gated channels. So these open and close when the transmembrane voltage changes. So that's something that we're going to discuss next. But if you have a change in the electrical voltage, then that will open and shut these voltage gated channels accordingly. And that's because the amino acids that make up these protein transport channels are charge sensitive. And like I said, they're involved in the propagation of action potentials, which we're going to discuss later on. Ligand gated channels. So these open and close when a ligand binds to the extracellular region of the channel. So you can see in these diagrams here, you've got the little red triangle that binds, and that allows the channel to open. Those ligands can be hormones, neurotransmitters, or other ions. But when we're talking about the nervous system, we're using neurotransmitters as the ligands. And finally, we've got leaky channels. So these just open and close intrinsically, which essentially means randomly. So there's nothing that controls when they open and close. And there are 25 times more potassium leaky channels than sodium leaky channels in the membrane, which means that cell membranes are much more permeable to potassium than they are to sodium. So each type of leaky channel can only let one different type of ion through. And there's a lot more potassium than sodium. So equilibrium potential. At equilibrium potential, there is no net movement of charged particles into or out of the cell. Sodium ions are 12 times more concentrated in the extracellular fluid than the intracellular fluid, and potassium ions are 40 times more concentrated in the intracellular fluid than the extracellular fluid. So if you think back to the diagram from a few slides ago, that's what I'm trying to describe here. So you might be wondering, why aren't these ions distributed evenly between the intracellular and extracellular fluid, considering I'm saying that we're at equilibrium here? And that's because we're going to discuss the movement of all the different ions now. So beginning with potassium ions will naturally flow out of the cell because they're 40 times more concentrated inside the cell. And as any type of substance would, it wants to move to the area of lower concentration. So naturally they will flow from inside to outside of the cell. However, the potassium ions are attracted back into the cell due to the organic protein anions, which cannot cross the plasma membrane, so remain inside the cell. So essentially you've got all of these negatively charged organic protein anions that are stuck inside the cell, which is making the inside of the cell highly negative. So that's attracting all of the potassium, which is positively charged, back into the cell while it's trying to leave the cell. So what I'm trying to say is the electrical attraction counteracts the concentration gradient, which results in this unequal distribution of ions at resting membrane potential. Next, we're discussing the movement of sodium. So sodium ions will naturally enter the cell because they're more concentrated outside the cell. However, they are electrically attracted back out of the cell by the chloride ions, which are found in the extracellular fluid. So again, that's the negative chloride ions attracting the positive sodium ions back out of the cell. This is because most cells are highly permeable to chloride, but they actually have no method of transporting it because we don't have any chloride leaky channels or any chloride channels that can actively transport chloride. So chloride ions don't influence the resting membrane potential when they move. Rather, their movement is influenced by the resting membrane potential. So they're going to stay outside the cells, and that's going to drag the sodium ions back out. Then we have the sodium-potassium pump. So the sodium-potassium pump counteracts the passive leakage of sodium and potassium ions, which is caused by the leaky channels. Sorry, that should be potassium. It's a positive ion, not a negative. The pump is a mode of active transport, which requires ATP to run. So as you can see in the diagram here to the right, it will transport 3 potassium ions into the cell, and at the same time, it will pump 3 sodium ions out of the cell and 2 potassium ions into the cell. So this is counteracting the sodium potassium that's going to be passively leaking out through those leaky channels, which open and close randomly. So the body can't control those. However, it can control the sodium potassium pump. So essentially that puts everything back to where it should be. And it's important to remember this ratio, the 3 sodium to 2 potassium. Now we have the Nernst equation. The Nernst equation is used to calculate the equilibrium potential for each ion. So we've got the formula here. So E ion is the equilibrium potential. Z is the ion's valence. So for all of the ions we've discussed today, sodium, chloride, potassium, they're all just a positive charge or a negative charge of 1. So that doesn't really matter. Then we've got concentration 0 or O, which is the outside concentration, and concentration I, which is the intracellular concentration. And then this table below, we've got all of those concentrations of the sodium, potassium, and chloride. So essentially, you'll just substitute these values in to find the equilibrium potential for any of these individual ions. So for the Nernst equation, you can only concentrate 1 ion at a time. So for example, if we wanted to know the equilibrium potential of potassium, we would put 1 because it has a valence of 1, then the concentrations of 5 and 150 for the outside and inside of the cell. And then once you do that equation, you get negative 90 millivolts. So that's the equilibrium potential of the potassium ions. Next, we've got the Goldman-Hodgkin-Katz equation. So this equation is used to find the overall membrane potential from the relative permeabilities and concentrations of each ion. So while the last equation was just finding the equilibrium potential for one individual ion, now we're finding the membrane potential for the entire cell altogether. And we've got the formula here. Yeah, actually on the next. This is straight from your notes really, because I didn't want to write this whole thing out again. And honestly, you don't really have to worry about this. If you want to do all the maths, you can put the numbers in, and this just explains how we calculate the equilibrium potential, the resting membrane potential to be negative 70 millivolts. But that's not a number that's going to change. So what I'm trying to say is you don't really need to use this formula, because this just explains why it's negative 70 millivolts. But you never need to calculate that on your own, because that's a given. So these two equations are really just more to explain how we get to these numbers, but this isn't something that you're going to need in an exam. So yeah, what I'm like, don't stress about these too much, because they're just to explain how we get to those numbers. All you need to remember is the numbers themselves. So negative 70 millivolts for the resting membrane potential. That's the most important part. Now, action potentials. So the learning objectives are the difference between a graded and an action potential, the concept of threshold and the properties of the ion channels involved, the events that return the membrane potential to the resting level after an action potential occurs, the concept of refractory periods and the two different types of refractory periods that occur after an action potential, and the role of ion pumps or transporters in recovery. So what is an action potential? Action potentials are electrical impulses which convey information down a neuron. They induce a change in the resting membrane potential of each axon until information has reached its target destination. So basically, they're the neuron's way of communicating with other neurons or other parts of the body. So there are a few different types of changes in the resting membrane potential. So if you look at this diagram to the right here, where I've put that red circle, that is the resting membrane potential, because that's at negative 70. So this straight part of the line, that's where your cells would normally be while they're just resting and there's no action potentials occurring. So they just stay at that membrane potential. Then we've got depolarization, which is our first stage. So depolarization is when the membrane becomes less negative than resting membrane potential. So that's a movement in the upwards direction towards 0 millivolts. So you can see that on the uptake of the graph here. Then we have repolarization. So that's when the membrane becomes more negative to return to resting membrane potential. So you can see that on the downstroke of the graph. And then hyperpolarization is when the membrane becomes more negative than resting membrane potential, or that's below negative 70 essentially. Graded and action potentials. So we have two different types of electrical signals, graded or action. Graded potentials are precursors to action potentials. So they occur at adjacent sites on the neuron, not at the same site. Essentially, a graded potential is where you have passive current flow, and passive current flow from the site of a graded potential will depolarize the adjacent membrane. And if this reaches threshold voltage, which is negative 55 millivolts, it will trigger an action potential. So basically you have a graded potential first. If that gets to threshold voltage, then it sets off an action potential. So just a little comparison between the two. Graded potentials are local, smaller changes in membrane potential. They're short distance signals and they spread bidirectionally, or if you imagine the site of the graded potential in the center here, the signals will travel both ways down a neuron. Whereas in action potentials, these are much faster and they're greater changes in the membrane potential. So if you saw on the graph before, they change from negative 70 millivolts up to about 30 millivolts, whereas graded potentials would be a much smaller difference than that. Action potentials travel much longer distances than graded potentials. However, they only spread in one direction. So if you have your action potential here, the electricity will only travel in this direction, not back that way as well. And they're all or nothing, which we will discuss later. But all or nothing is an important buzzword in exams, because graded potentials aren't all or nothing, but action potentials are. So voltage-gated sodium channels, they have 3 states, as you can see in the diagram. We've got open, closed, and inactive. So sodium channels allow sodium ions to enter the cell down their concentration gradient. Also, these are one of the types, if you think back to the previous slides where I said there were 4 different types of transport channels, voltage-gated channels were one of them. So they allow sodium ions to enter the cell down their concentration gradient. They open and close rapidly. They're generally closed at resting membrane potential, because at resting membrane potential, the only way that sodium ions are moving is through the leaky channels and the sodium-potassium pump. And when sodium channels are inactivated, they will not open regardless of the stimulus, whereas when they're closed, they will open when there's stimulus. Then we have voltage-gated potassium channels. So these only have 2 states, open and closed. They can't be inactivated. They allow potassium ions to exit the cell, again, down their concentration gradient. And these open and close very slowly compared to the sodium channels. And again, they're closed at resting membrane potential. So those 2 types of voltage-gated channels, they aren't usually used when the cell is just at its normal resting membrane potential. These only come into play when an action potential occurs. So how do action potentials occur? Firstly, you've got your graded potential, which causes the membrane to reach threshold potential. So if you look at the graph here, you can see that -70, you've got that straight line, which is just resting membrane potential. That's how the cell is in its normal state. Then at -55, that's the threshold. So the graded potential is going to do the first part of the work. It's going to get the cell to -55, and then that's when an action potential can take off. So we have our first phase, which is depolarization, where the membrane potential becomes more positive, less negative. So in the first stage, the voltage-gated sodium channels will open and sodium ions will enter the cell. And this allows the cell to depolarize because sodium ions are obviously positive ions. So because you've got a lot of positive ions flooding into the cell, that will make it less negative. Then right up at the top, the peak of the graph, that's peak depolarization, which is positive 35 millivolts, anywhere between 30 to 35. Then we enter the repolarization phase. So that's when the sodium channels close and the potassium channels open. The potassium channels will open, allowing potassium ions to leave the cell. Because potassium ions are also positive and they're leaving the cell, that's going to make the cell back down to negative again. So that's our downstroke. And then as potassium channels are slower to close than sodium channels, like I said on the previous slide, sometimes you can have a little bit too much potassium leaving the cell just because they're slower to shut, so a little bit too much gets out. And that causes hyperpolarization. So that's where it becomes more negative than resting membrane potential. And you can see that on the graph where it has a little bit that goes under the line. After the action potential, sodium and potassium are returned to their original concentrations by the sodium-potassium pump. So that's going to pump the sodium back in and the potassium back in and the sodium back out of the cell. So that's just how it gets back to normal. So a little bit more about threshold voltage. So at threshold voltage, enough voltage-gated sodium channels are open to facilitate the sodium influx. Once threshold voltage is reached, anything above this will not create a larger action potential. And this is the meaning of all or nothing. So if you look at the graph, the little diagram to the right here, you can see in the top part, you've got your sub-threshold potentials. So these are graded potentials which have occurred, but they're not strong enough to reach threshold potential. So you're not going to have an action potential. Then you can see the action potentials. So suddenly threshold stimulus is increasing, which means that we've reached threshold potential, but all of your action potentials are the same strength. So if you look at the bottom graph, that's representing the threshold stimulus. So you can see even though the threshold stimulus increases, the action potentials stay the same size. They can't get any bigger once you reach threshold potential. Refractory periods. So this is the time after an action potential where a second action potential cannot occur. And the reason we have the refractory period is because it forces action potentials to travel in one direction, because the sodium channels in the previous sections of an axon will be inactivated. So if you think about an action potential occurring here, it travels in one way, and then the sodium channels in this direction are going to become inactive after the action potential is over. So then when you get a second action potential occurring here, it can't travel back in this direction because these sodium channels here are still inactive. So it forces it to only travel in one direction. So we've got two different types of refractory periods, the absolute refractory period and the relative refractory period. So in the absolute period, all of the sodium channels that were involved are either open or inactive, so no action potential is possible. In the relative refractory period, most of the sodium channels have returned back to their closed state instead of their inactive state, so they can be re-stimulated. However, some potassium channels are still open in the relative refractory period, which means they're still letting some potassium leave the cell. This means that a larger threshold is required to stimulate a response. And that's it from me.
Okay, so I'm going to be taking you guys through the nerves prac, and then also a little bit about synaptic transmission. So starting with the nerves prac, it was a bit of a random prac. There's just it's kind of just like background to nerves and all that sort of stuff. So I'll just take you through what we learnt in our nerves prac. So to start off with, we have some helpful terminology. If we go to the next slide. Yeah. So just some things that you need to know. Afferent, when we describe something as afferent, we mean it's going towards the CNS, and efferent is away from the CNS. Ventral is used to describe the front, and dorsal is used to describe the rear. Your ganglion is your cell nuclei in your peripheral nervous system, and your nuclei is also your cell nuclei, but it's your nuclei in the central nervous system. Proximal is what you refer to as something close to the body center, and distal is something that's further from the body center. Ipsilateral is on the same side, and contralateral is on the other side. So this is just something helpful for you to keep in mind, and you can return to it later on when we go through in the weeks to come some more nerve stuff. So next slide. Just a little bit about nervous system embryology. You really don't need to know that much at all. It's really just a short overview because you'll go into it a lot more in year 2. But pretty much all you need to know is this purple thing is a neural plate, and that's what our nervous system develops from. So the neural plate folds up and it forms the neural tube. If you can see that in the third image, it's got a little flow chart, and the third image in the flow chart is your neural tube, and that forms your brain and your spinal cord. And then the neural crest, if you see that as well, those little like green dots, that migrates away from the neural tube, and it forms most of your peripheral nervous system and lots of other tissues. So you really don't need to know much about the embryology at this point in time. So just a little bit about nervous system divisions. So you've got your central nervous system and your peripheral nervous system. So your central nervous system is pretty much the main control center of yourself. It's the brain and your spinal cord, and that just comprises of the brain and the spinal cord. So it doesn't have any subdivisions that you need to know of at the moment. It's just the central nervous system. And then you've got the peripheral nervous system, which has your peripheral nerves, and it's pretty much how your body communicates to your brain and your spinal cord. So within the peripheral nervous system, we have the sensory division and the motor division. So your sensory division is how, like, say you touch your hand to a burning hot plate. That will be sensory nerves, which will be bringing the pain back up to your brain to communicate that. So it's composed of sensory neurons. So that's pretty much peripheral to CNS. And then your motor division is from the CNS to effector organs, which could be muscles, it could be glands, something in response to your what your sensory neurons have already passed on to you. So, and then within the motor division, we've got your autonomic nervous system and your somatic nervous system. So in your autonomic nervous system, it's responsible for controlling involuntary responses, and your somatic nervous system is controlling voluntary movement. So your somatic nervous system is things like muscles. So, you choose to move a muscle, you choose to do something else, but your autonomic nervous system is something that you don't choose to do. So, and then one more further division, in your autonomic nervous system, we have the sympathetic divisions and the parasympathetic divisions. So your parasympathetic is often referred to as the rest and digest, and your sympathetic is referred to as your fight or flight responses. And I'll just go a bit into sympathetic versus parasympathetic next. So this is just, you'll go a lot more into this in semester 2 for sure. So just a quick overview of your sympathetic and parasympathetic nervous systems. So sympathetic is your fight or flight. So if you think about what happens when you're in a situation where you need to run, you've got a faster heartbeat, you've got relaxed airways so you can get more oxygen into your lungs. You've got inhibition of a lot of your organs because it needs to, like, the energy needs to be devoted to you getting out of a scary situation pretty much. Like secretion of adrenaline and hormones like that. Your pupils become dilated, but when you're in a when you're in a rest and digest, so just your normal resting position, you've got your parasympathetic nerves which are activated. So like you salivate, your heartbeat slows, your airways are constricted, you don't need to take massive breaths anymore. Your organs are like back to working like they usually were, and your pupils are constricted. So you will go over this a lot more next semester, especially when you're talking about how drugs affect various parts of the nervous system. So don't worry about that too much now. Just know it's kind of fight and flight and rest and digest for your parasympathetic and sympathetic, but the other way around. Okay, so now a little bit about the cells of the nervous system. So first of all, we've got nerve cells, which are known as neurons. So neurons are pretty much just cells that communicate with other cells, whether that be communicating neuron to neuron or communicating to your muscle or to glands. But a neuron is composed of a cell body, dendrites, and an axon. The cell body is usually pretty compact, and the axon and dendrites are filaments that extrude from the cell body. So I wonder if we go on to the next slide, and I'll just show you what it looks like. Okay, so here's what your neuron pretty much looks like. If you see, you can see the dendrites, which receive the signal. You've got your cell body, otherwise known as a soma, there in the center. Then you've got your axon, and that's responsible for transmitting the signal. So you've got your axon hillock, which is where the axon separates from the cell body. And the axon can actually be really long. Sometimes it can be up to 1 meter long in some humans, like in some cells in humans. So you're not always thinking about something very, very small. If you have a look at the end, you've got your axon terminals, and that's how the signals are sent to the next cell, and then you've got your synapse. So if you see the synaptic end bulbs, just past that you've got your synapse, which is the gap between the two cells, but I'll talk more about that in synaptic transmission next. Next up, we've got neurons are generally classified by their structure and their function. So structure, there's 4 different types of neurons you can have. You can have a unipolar neuron, which is just the cell body with one extension. They're not in humans, so they're very irrelevant. Just know that they exist pretty much. They're found in insects, actually. Bipolar is just a soma, one axon and one dendrite. Multipolar, you've got one axon and multiple dendrites, so you find that in your central nervous system. And then pseudo-unipolar, which is one axon and one dendrite, or one sensory receptor. So most sensory nerves are made of this. Next up, we've got, in terms of function, you can classify them. So you've got your sensory neurons, as we were talking about before. They take information from receptors to your CNS. Your motor neurons are the CNS back to effector organs, so whether that be muscles or glands. And then an interneuron is just responsible for connecting your sensory and your motor neurons together. Next slide is just the different types of neurons. So if you see there's a unipolar neuron, top left, you will not need to know that. That's an insect neuron. Then you've got your bipolar neuron, which you find in the retina of the eye, but you'll probably cover that when you do the eye later. Your multipolar neurons, so they're the most common ones, and then your pseudo-unipolar neurons. And then if you see here, you've got your motor neuron, your interneurons, and your sensory neurons. Next slide. So your second type of cells are your supporting cells, known as glial cells. So they pretty much hold the neurons in place, they provide insulation, and they're also responsible for supplying neurons with any nutrients, any oxygen, and like clearing out any debris from the cell that's needed. So some different types of glial cells that we have are, in your central nervous system, you've got astrocytes. This is really fuzzy. Oh no, it's better now. Okay, so you've got astrocytes. They're responsible for maintaining the blood-brain barrier. They provide structural support. They regulate any concentrations necessary within the neurons, and they also form scar tissue after injury. Then you've got your ependymal cells, which line your ventricles in your brain, and then also the central canal and the spinal cord, and they're very important in the maintenance of the cerebrospinal fluid. Then you've got your oligodendrocytes, which myelinate the CNS axons, and they also provide framework. And the microglia, which are important for removal of cell debris, wastes, and any pathogens. So that's in your CNS. And then you've got two other types of cells which you find in your PNS. So you've got your satellite cells, which surround the neuron cell bodies in the ganglia, and they pretty much regulate a lot of important levels in these ganglia. So that's including your oxygen, carbon dioxide, nutrient, and neurotransmitter levels. Because if we didn't have satellite cells, then those levels would be really mucked up, and that would majorly affect how impulses are transmitted along the PNS. So then we've also got Schwann cells, which are actually surround all axons in the PNS, and they're responsible for the myelination of peripheral axons. So they're very important because they can increase the speed at which nerve impulses can be transmitted. And on the next slide, I'm just going to quickly differentiate between white and grey matter for you. So white and grey matter is pretty much, white matter is in the brain, you'll see that, and that is your axons, and grey matter is your neuronal cell bodies. And white matter is actually white because of the Schwann cells, which we covered earlier, which provide myelination and increase the speed. So now a little bit on synaptic transmission. So a synapse is pretty much a specialized junction where one part of a neuron will contact and communicate with another neuronal cell. So this could be a muscle cell or a glandular cell, like we were describing earlier. So these junctions are described as pre or post-synaptic. If you have a look at this image down there, you can see a synapse. So you can see where one neuron, the axon terminals, are synapsing onto the next dendrites. So they're very important for communication from one cell to another. And then this communication is called synaptic transmission. So synaptic transmission can either occur electrically or chemically. So some quick differences. Chemical synapse is a lot slower than electrical synapse. And chemical synapse actually uses neurotransmitters, whereas electrical synapse does not use neurotransmitters. So neurotransmitters are these molecules which I'll cover later, but electrical synapse doesn't use neurotransmitters, only uses gap junctions. So chemical synapses are important for amplifying information, so spreading it to more effector organs, whereas electrical synapse just transmit information. In addition, chemical synapses can go to neuron to other cell or to neurons, but electrical synapses are only really neuron to neuron. So if we go on a little bit and have a look at our gap junctions and our neurotransmitters, this is just a quick overview of some differences. So your neuron looks very similar still, but if you have a look at your presynaptic neuron and your postsynaptic neuron in image A, you can see gap junctions. And then if we zoom in on that a little bit, you can see these gap junction channels, which are actually providing the flow of ions through. So that's why it's so much faster, because there's already these specialized transmembrane channels, protein channels, which exist to facilitate the flow of ions. Whereas if we switch on to the second image, which is our chemical synapse, you can see that there aren't these channels anymore, and there's actually like these little vesicles which have to fuse with the presynaptic membrane and then release neurotransmitters to pass the message on. And there's still postsynaptic channels, but they're just not connected like they are in electric synapse. So it just takes a lot longer because these neurotransmitters have to diffuse across the synaptic cleft and then go through the ions and then like hit the postsynaptic cell. So next slide, just a little bit on electrical synapse. So electric synapse is important because it allows the direct transfer of ionic current from one cell to the next. So it occurs at gap junctions like you saw in the image before. So gap junctions are specialized sites that allow the direct transfer of ionic current from one cell to the next. These two ion channels are electrically coupled, so they kind of like stick together, and they allow actually electrical current to go back and forward, which is an interesting property, and the transfer is very fast. So it's pretty much a fail-safe method of transferring this ionic current. It's very important in the heart, because if you had chemical transmission in the heart, it would be very open to error, and you don't want that in the heart. You want something that allows all your heart cells to contract at the same time so you can get that blood pumped around your body, because imagine if you didn't have that, then you'd be having heart cells contracting at random times of the day or at random times of the minute, and it just wouldn't allow you to pump blood around the body. So it's kind of life-saving that we have electrical synapses in our body. So a chemical synapse is the main mechanism of synaptic transmission. So if you have a look, we've got our synaptic cleft in this image. That's the space. It's not connected. So unlike an electrical synapse, the two presynaptic and postsynaptic neurons aren't electrically coupled. So we've got our voltage-gated Ca2 plus channels, which are found all around the body. Different structures do exist to these channels, but not all functions are known. Sorry, it's my dogs. So the synaptic cleft is 20 to 50 nanometers, and it separates the two membranes. And this is actually quite large, because even though in gap junctions there is a small gap, this is like 10 times smaller than that of a synaptic cleft. So the synaptic cleft is actually decently large. And the cleft is filled with a matrix of extracellular protein. So even though they're not connected to each other, this extracellular matrix does allow the presynaptic and postsynaptic neurons to stay pretty much connected, because after all, they still have to be pretty close. Like 20 to 50 nanometers is still pretty close. So I'll just go over the steps quickly. So we have neurotransmission at a chemical synapse begins with the arrival of an action potential at the presynaptic axon terminal. So if you see step 1 on that image, so that's your action potential, which has just reached the terminal. So then we have our voltage-gated calcium channels, which open, and the calcium enters the axon terminal by its electrochemical gradient. So if you have a look at step 2, we've got our calcium, which is entering the axon terminal. So then calcium is acting as an intracellular messenger to bind with the proteins in the axon terminal, which then results in the formation of these synaptic vesicles. So these, if you see these little yellow dots, which are termed the synaptic vesicles, they're what's being formed. So then the calcium is removed from the terminal because it's not needed anymore. It's already done all the signaling it needs, and we've got our synaptic vesicles ready to diffuse across the membrane. So then if you have a look, we've got our neurotransmitters, which are diffusing across the synaptic cleft, and they're binding to their specific receptors on the other side of the membrane, on the postsynaptic membrane. And this is then opening up the ion channels. So if you have a look, we've got our neurotransmitter binds to postsynaptic receptors, and then this is opening up the channels. So our vesicles are then able to diffuse through to the postsynaptic membrane, and it results in a new AP, like a new action potential being generated in the next cell. So it's a process that repeats itself until it comes to its target organ or target cell or whatever it needs to finish off doing. So there are 3 ways to remove neurotransmitters. One of them is reuptake. So reuptake, the neurotransmitters are both taken back up by the presynaptic terminal and they're reused. Otherwise, you can have degradation, in which the neurotransmitters are broken down by enzymes, and this is often on the postsynaptic membrane. So enzymes lying kind of past this synaptic cleft on the postsynaptic membrane can break down the neurotransmitters. Or they can just diffuse away from the synapse. And that's where some of the glial cells are important in clearing away those neurotransmitters and debris so they don't clog up on the synapse. Okay, next slide. So, neurotransmitter action. So neurotransmitters, when they bind to their postsynaptic membrane, they open up ion channels, which then allows greater potentials, and they actually undergo conformational changes when they bind to these open channels. So you've got 2 different types of receptors. So you've got your ionotropic receptors and your metabotropic receptors. Your ionotropic receptors lead immediately to graded potentials happening, but your metabotropic receptors are actually, they open up the ion channels, if that makes sense. I'll go over that a bit later, but I've already talked about the termination of the neurotransmitter. And synaptic delay. So chemical transmission is a lot slower than electrical. It takes about probably between 3 times and 50 times the length of time that electrical transmission takes. And this pretty much just reflects the time needed for the neurotransmitter release, the diffusion across the cleft, and to bind to receptors, because there's a lot more steps in chemical transmission than there is in electrical transmission. So synaptic delay is actually the rate limiting step of neural transmission, because electrical transmission is a lot faster. Okay, next slide. I think I go into more depth on neurotransmitters. Yeah, so neurotransmitters are pretty much always there in your presynaptic neuron, and the release of calcium then allows the neurotransmitters to be released in the vesicles past the membrane. So these neurotransmitters, each neurotransmitter has a specific receptor, and depending on what the neurotransmitter is, there'll be the receptor for that on the postsynaptic cell. So a neurotransmitter might be excitatory or inhibitory. So pretty much the neurotransmitter might make an action happen, or it might make an action stop. So you can have neurotransmitters can produce different types of responses on different cells. So a neuron might synthesize and release 3 neurotransmitters for a certain action. It might release just 1. It really depends on what action is needed. So that means that you need to be your body needs to very carefully regulate how many neurotransmitters it releases and how much there is present, which is why the glial cells are also important again, for regulating how much neurotransmitter you kind of just have swimming around in there. Yeah, and pretty much neurotransmitters are synthesized in different ways. So you have some made in your cell body, you have some made in your cytosol, which are then packaged into vesicles later on. Okay, so IPSPs and EPSPs. If you have a look at these diagrams, an EPSP is an excitatory postsynaptic potential. So that's when you have a greater potential after the neurotransmitters have diffused across and they cause a greater potential. So if you have a look here, it's depolarized the neuron. The neuron's now less negative and it brings it closer to the threshold. So if this keeps happening, if you have lots of EPSPs in a row, then your neuron's going to be depolarized past the threshold and you get an action potential, like Meg covered earlier. And then inhibitory postsynaptic potential hyperpolarizes the postsynaptic neuron. So that pretty much lowers the likelihood of reaching the threshold. If you have a look here, it's making it more negative. So you really are much less likely to get an action potential after an IPSP. And that might be what your body wants. So the body pretty much has to control which neurons are released, because some neurons will evoke an EPSP and some will only do an IPSP. Next slide. So your types of receptors that you can have, you've got your ionotropic and your metabotropic. So ionotropic receptors are an ion channel themselves. So pretty much they just, when they're activated, they open channels which allow your ions to flow in into the cell and generate the new action potential. So it's generally pretty fast postsynaptic effects because the ions can flow straight through these channels, but the effects are also very short-lived. So they only live for about a few milliseconds. They're good to very quickly spread on the next action potential. And metabotropic are slightly different because they need an intermediate molecule before these ion channels are opened. So these intermediate molecules are called G proteins. And once these molecules are opened and activated, then a series of intracellular events are triggered. So this may result in ion channels opening, but there's also the second messenger chemicals like the G proteins, which have to act before the ion channels open. So generally these are much, much slower responses. So you can have hundreds of milliseconds, minutes longer even, but they amplify the response of the initial neurotransmitter. So they're often found on effector organs rather than just passing the action potential down the chain of neurotransmitters. Next slide. I think I'm almost at the end. Clinical application. This is just a very short one, but pretty much synaptic transmission is very important for a lot of psychoactive drugs. So you'll cover them a lot when you do psych later on, year 4 onwards. So antidepressants actually are selective serotonin reuptake inhibitors. So they're inhibitory drugs, and they prevent the serotonin from being reuptaken into the presynaptic neuron. So that pretty much means that your serotonin levels in the synaptic cleft are a lot higher. So that's just a cool, a kind of cool way to think about it in a more clinical way, rather than just thinking about, presynaptic, postsynaptic. You can start to think about how drugs can kind of be used, use these mechanisms. And this will be covered so much more next semester. So even if you don't, there's a lot of specifics that I've covered today, so many specifics. But I think it's just a really important thing to take out, that drugs will use this a lot, and you've still got the basics. Like, you can come back and look at the basics when you're looking in revision period and swap back, all that sort of stuff. And I don't expect you to understand it all, because, to be honest, like, hardly I didn't understand it when I first learned it, because there's just so much that you cover with neurotransmitters and everything. But just knowing the basics, that, yeah, antidepressants actually like make use of this neuro junction as well. I think that's the end. This is just a handy table. I'll just leave that one on the slides for you guys to look at in your own time. And I'm pretty sure that is my part done. Yeah. Over to you, Ethan.
Easy, thanks, mate. So I'm going to be talking about spinal cord organization. And this was one of the more challenging lectures, sort of at the back end when tracks come into play. So, you know, don't panic too much when whoever the lecturer is, it might be Michael, starts going through the tracks. But Teach Me Anatomy has, you know, really good pages on each of them. So that's definitely a good read if you're, you know, struggling to understand that sort of content. But we're just going to overview pretty much what the spinal cord is, how it all works, and there'll be different sections to what we go through in terms of whether the content's really important or, you know, a bit niche. So reflexes, often, you know, we use them in our daily life. Like if you touch something that's too hot, your hand will retract, you know, pretty much without you thinking about it. And so, you know, different types of reflexes, we can have monosynaptic, mono meaning one, poly meaning many. So monosynaptic just means one synapse, right? And polysynaptic means many synapses. So with our monosynaptic reflex, we've got a afferent nerve or a sensory nerve that takes the signal into the central nervous system, often the spinal cord, and then that will synapse onto an effector, you know, or motor nerve, which will actually initiate a action. Whereas with polysynaptic reflexes, we have interneurons between the sensory and the motor nerves. And, you know, these can trigger the actual action while also sending signals up to the CNS, up to the brain. So you can actually, you know, process what's going on. So this whole process of, you know, a receptor on your body being triggered, afferent nerve takes it to the CNS, an efferent nerve comes down and, you know, activates a muscle causing a response. That's called a reflex arc. And the sort of time it takes between the initial signal and your response, you know, it's not instantaneous, right? There's a little bit of a delay, and that delay is called reflex latency. So that's caused by, like, it takes time to actually conduct the signals to, you know, process it all. And those, you know, correspond to what we call a reflex latency, right, and a little bit of a delay. Just a reiteration that, like, afferent nerves go to the CNS, they're your sensory nerves, and efferent nerves, your motor nerves, go away from the CNS. So I may touch on this before. These are just the nervous systems of the body. We have our peripheral nervous system, which is your sort of anything outside the brain and spinal cord. That is split up into sort of voluntary, somatic nervous system, and involuntary autonomic nervous systems. And you also have cranial nerves arising from the brain, although they're not so important at this stage. So your autonomic nervous system will regulate your organs, your internal environment, and it's all unconscious. So things like homeostasis, that's all autonomic. And, you know, may mention the fight or flight response. And that's just what this slide here is talking about. So the actual spinal cord has different segments to it. And basically the actual vertebrae at different segments are different, and they'll innervate different parts of the body, right? So the spinal cord is protected by all these vertebrae, connects the brain with the body, and it's involved in both your somatic reflexes and your autonomic reflexes. An example of an autonomic reflex is, for example, like when you eat food, your stomach expands, that sends signals and, you know, activates some digestive processes. So we can have both, like, voluntary and involuntary, or somatic and autonomic reflexes, right? The actual spinal cord divisions are split into cervical, thoracic, lumbar, sacral, and coccygeal. Really, like, the first four are the ones you see quite a lot of. And there's 8 cervical vertebrae, 12 thoracic vertebrae, 8 cervical roots, 12 thoracic nerve roots, 5 lumbar nerve roots, and 5 sacral nerve roots. So basically that just means like at each sort of vertebrae, there are different nerves that come out, and these nerves correspond to, the vertebrae that you see here. At the level of like the L1, L2 vertebrae, the spinal cord stops becoming an actual cord and just becomes sort of a bundle of nerves. That's not so important now, but that'll have some relevance in semester 2 when we think about like when we want to put in needles into the spine. Okay, but the key takeaway is there's just different vertebrae that you see, different categories, and sort of at each vertebral level, there are some nerves that come out from either side and go to various structures. So again, white matter and gray matter is something that's been touched on today, but this is a cross section of the spinal cord. Basically, you sort of cut it transversely. So, the spinal cord runs up. It's like you take a cut maybe halfway through. This is what like the inside looks like if you're looking top down. And so we have gray matter, which is your cell bodies and your unmyelinated axons, and then your white matter, which is your tracks, like your myelinated and unmyelinated axons going out. Okay, so areas that require more sensory and motor control, your limbs with your finger muscles and things like that, they'll have more cell bodies, and so you'll get a greater proportion of gray matter. But this is really like the more important stuff, right? So this is a very common example that you'll see. Michelle Lazarus does a good lightboard video on it. If you've got access to that, it'll be in some strange middle folder. But basically, there'll be a few workshops that you do where you've got to sort of describe what's going on and pretty much label this diagram. So again, reiterating that dorsal means sort of towards the back or posterior, and ventral means sort of towards the front, or anterior is another word for that. Okay, so pretty much your sensory fibers will come and enter the spinal cord via the dorsal root ganglion. This is this little bulge that you see on the side here. So the sensory fibers come in and into the spinal cord via the dorsal side, so from the back, and they have their cell bodies in this dorsal root ganglion. Ganglion is just a cluster of sort of cell bodies, and it's notable that these are the pseudounipolar neurons. So it's one neuron, the cell body's here, and then the tract continues on to the spinal cord, even though the cell body is in this DRG, right? So sensory nerves come in, have the cell body here, and then they'll synapse in the spinal cord. Whereas motor neurons will have their cell body actually in the spinal cord, and they'll just run out through this area here, right? So we call this part where your sensory signals travel, we call that the dorsal root. So it's on the dorsal side. So we call this bit the dorsal root, and similarly, this is the ventral root. So the ventral root, only motor signals, dorsal root, only sensory signals. They'll eventually merge to create a mixed spinal nerve, carrying both sensory and motor signals, and that'll eventually split up into what we call a dorsal ramus and a ventral ramus. Dorsal ramus just goes to the back, ventral ramus goes to the front, but they're both mixed nerves. So that's key. So before it becomes a mixed spinal nerve, your sensory and motor, they're separate, but so away from the spinal cord, everything's mixed, right? So again, like pretty important diagram to know. If you can just remember that, mixed spinal nerves come in, sensory nerves have their cell body here in the dorsal root ganglion. They travel via the dorsal root into the spinal cord, where they'll eventually synapse and project neurons upwards to the CNS, and then your neurons from the CNS, the actual effector neuron has its cell body in this ventral horn, and it will come out via the ventral root and then form a mixed spinal nerve, and then it might go through the dorsal ramus or the ventral ramus. Okay, so again, this is just a horizontal cut section. At every level of the spine, you'll have these ventral and dorsal roots coming out of it, and so we get a whole bunch of nerves coming out of the spinal cord. Now we get into the tricky stuff, which is tracks, right? So the specific pathways, from sensors from receptors up to the brain, and then from the brain actually to motor organs, right? So ascending tracks are your sensory pathways. They move from the peripheral nervous system to the central nervous system, and descending tracks go the other way. So the nerves start, in the brain and then go downwards, and actually result in effector action. So when we talk about ascending tracks, there's generally three neurons, right? In all of these tracks, there'll be 3 neurons, and that's the primary or first order neurons, the second order neurons, and the third order neurons, or primary, secondary, tertiary, right? So your primary neurons will connect the actual peripheral receptor to the spinal cord, and they're the ones that have their cell body in the dorsal root ganglion, right? Then, so like let's say someone like hits you on the hand, receptors pick up that signal, they trigger a nerve, that nerve will come and enter the spine. That's your primary neuron. Once the nerve's in the spine, it synapses onto a second order neuron, and that will move the signal up the spine into the thalamus, which is in your brain. And third order neurons will begin in the thalamus and transmit signals to the relevant brain center, which might be the primary sensory cortex. So really, like your first order neuron comes from the periphery, goes into the spine. Then in the spine, it will synapse onto a second order neuron, which will go up to the brain and up into the thalamus. And then your third order neuron is in the thalamus and will go to the relevant center. So that's ascending tracks, 3 neurons. Descending tracks are a little bit more simple. There's just 2 neurons, upper and lower motor neurons. So the brain sends out a upper motor neuron that will come down to the spine to the required level. Then at that level of the spine, it will synapse in the ventral horn. So if we go back here in this ventral horn, it will synapse. So there'll be a nerve here onto this lower motor neuron, which will go and innervate the muscle. So ascending, 3 neurons, primary, secondary, tertiary. Descending, 2 neurons, upper and lower motor neurons. Okay. So basically different tracks carry different signals, as you see down the bottom here. We're going to talk about the dorsal columns and the spinothalamic tract and the corticospinal tract as well. But yeah, it can be useful just to know like what tracks carry what signals, and then a brief overview of how the actual tracks work. It might seem a little bit daunting, but if you can just remember like ascending tracks, primary, secondary, tertiary neurons, and then I find that memorizing the picture or understanding how the picture works is generally helpful. So the first track that you need to know is the dorsal column medial lemniscus pathway. Really, like what those words mean doesn't matter. Just call this DCML pathway. Dorsal just means sort of sitting at the back, like the dorsal root. So if we actually look at the spine, like this tract, is situated in the posterior, the dorsal regions, right? So this tract is going to transmit signals of fine touch, pressure, vibration, proprioception, which is like if you close your eyes, you sort of know where your body parts are. That's proprioception. So it's like your sense of where everything is, where all your body parts are. So, I'd suggest just remember that this pathway carries these signals. Okay, and then the overall sort of map of the nerves. So following on from what I said before, we have first order neurons that come from the receptors. They enter the spinal cord and they'll actually travel up into the medulla, which is in the brainstem, right? So rather than they don't synapse straight away, they enter the spinal cord and then go up a little bit. Then they synapse onto second order neurons, still in the spinal cord, and these switch over to the opposite side of the body. So you may have heard the left side of the brain controls the right side of the body. And this is sort of relevant to that, right? So any signals from the right will switch over, which is called decussating, to the left side of the body. So your first order neuron enters the spinal cord, moves up into the medulla, part of the brainstem. Then it will synapse into second order neurons, which switch to the opposite side of the body, and then they'll take it up into the thalamus. And The good thing about tertiary third order neurons is that they always like start in the thalamus and then pretty much go to the primary sensory cortex, right? So that's a common feature. So you pretty much need to remember with ascending tracks, like where this first synapse is and where the decussation is. So where the first order synapse is onto the second order neuron and where the actual tract switches to the opposite side of the body, right? So again, try and use the pictures. Then we have the spinothalamic tract, and this transmits poorly localized or crude touch, pressure, pain, and temperature. And again, it's first, second, and third order neurons. So our first order neurons, conversely, will just go into the spine and synapse straight away, right? And also the decussation happens straight away as well. So first order neuron comes into the spine, synapses straight away, second order neuron comes in, crosses over, and then just goes straight up to the thalamus. So and this one might be a little bit easier to remember. Quite literally, enters the spine, synapses, decussates, goes up to the thalamus. And again, it's the same thing of third order neuron, starts in the thalamus, goes to the primary sensory cortex. And that's the spinothalamic tract. So again, probably memorize the actual signals that are being transmitted, and then where the synapse is and where the decussation is. So those are the two ascending tracts you need to know. Then the descending tract you need to know is probably the corticospinal tract. And this corticospinal tract is part of the pyramidal tracts. So I'll just briefly talk about them. So the pyramidal tracts are your descending tracts that controls your muscles. And they consist of your corticospinal tract, which controls the body, and the corticobulbar tract, which controls the face. So different tracts control the body and the face. The corticobulbar tract starts at the pyramids in the medulla, part of the brainstem, and innervates these cranial nerves that innervate your face. And there are 4 descending tracts that are outside of the pyramidal tract. But basically, how this pyramidal tract works is it controls your muscles. A part of it controls the face, that's the corticobulbar tract. A part of it controls the body, which is the corticospinal tract, which is what we're going to look at. This is the corticobulbar tract, where your neuron, and this is your upper motor neuron from the brain, comes and synapses in the medulla, and that will send out a lower motor neuron to the face muscles. But that's not so important. What's more important is this corticospinal tract. And just remember, now we are descending. So we're starting up here and we're leaving down here with upper motor neurons and lower motor neurons. So our upper motor neuron will begin in the cerebral cortex, which pretty much controls your movement. Then you don't really need to know about internal capsule and medulla. But what you do need to know is the tract sort of splits up. So it starts here in the cerebral cortex, comes down to the medulla, and then some of the fibers, and 80%, will decussate, so switch over, and then come down, and wherever the motor signal needs to leave the spinal cord, then it will synapse and leave as a lower motor neuron. This is one upper motor neuron, starts in the cerebral cortex. It will come, most of the fibers will switch over at the medulla. The remaining 20% just don't decussate, and they'll synapse in the cervical or upper thoracic levels of the ventral horn. So they don't decussate, and they'll come out as a lower motor neuron somewhere else. But really what's important is like starts in cerebral cortex, splits in the medulla, and it will sort of, the synapse is at the level of where you actually need the muscle action, right? So it's really these 3 tracks that you need to know. All the other tracks are not really required, right? So we call the part of this corticospinal tract that actually switches over at the level of the medulla, and that's the lateral corticospinal tract, and that will sort of control your peripheral muscles. Whereas this anterior spinal tract, which is the remainder, that will control your trunk muscles or your sort of chest midline sort of muscles. So not the limbs. Okay. So yeah, those 3 tracks are all you need to know, and that's really the synthesized version of this lecture. Last year we had Michael for it, went through fairly quickly. So yeah, don't be afraid to have a read over, but I highly recommend the Teach Me Anatomy pages for these tracks, because they explain it quite well and they're good to read in your own time. So that's all from us today. Thanks very much for coming. If you have any questions, feel free to let us know in the chat. Yeah, hopefully you have a great week, and we'll hopefully see you next week for some more PSPirates.
Thanks, everyone.
Thanks for coming, guys.
Thank you.